So great pleasure now to introduce our second speaker, Dr. Paul Brennan, who's the Clinical Director of the Northern Genetics Service, to tell us much more about the 100,000 Genome Project. Thank you.
What a small man he is. There we are. So I'm not Scottish, I'm from Kent, so the accent's going to be different, I'm afraid. I want to talk to you about this. This is very exciting, but there's a lot of terminology involved, so I'm going to spend the first few minutes defining some terms for you, if that's all right. So I work I'm a clinician, I'm a clinical geneticist, I'm a specialist in inherited conditions. And currently I'm the clinical director of the Northern Genetics Service, which is based down at the Centre for Life, just down the road. Earlier this year I became the clinical lead for a new structure. This is a new NHS structure, and we now in this region have a genomic medicine centre, and I hope that I can explain what one of those is as we go through this. So the first question that lots of people ask me, what actually is a genome? So we're having a 100,000 genomes project, but what actually is a genome in the first place? And the simple answer is the genome is all of your DNA. Okay, so it's the DNA that you inherit from your parents. And your genes only account for 2% of that DNA, and the rest of your DNA is something else. Okay? It's actually derived from a German botanist who came up with the word genome. It's derived from the German word for gene, which I guess is pronounced gen, and the German word, or part of the German word, for chromosome. And that's where we get the word genome. So it's a bit made up, actually. And it's made up by a German botanist. Quite interesting. Just to remind you, this is the problem. Yeah, okay. Can you all hear me, actually? Is that all right? Up a bit more? Higher? Okay. So I'm going to use, what about that? That's better, isn't it? Okay. I'm going to use the word chromosome probably once or twice. So if you open a cell and open the nucleus within the cell, there are structures called chromosomes. And if you open a chromosome up, they are beautiful structures composed of DNA, which has been almost like a piece of cotton rolled up into a ball over and over again. And it's a way of packaging a very long DNA molecule. Why is Mary Berry there? Well, she's groovy, obviously. If you think about a chromosome like a recipe book, okay, and that's the way to think about the chromosome. So the genes within that book are the recipes, okay? That's the way to think about it. So 2% of Mary Berry's book would be useful recipes, and 98% would be unuseful stuff, okay? Pictures and adverts and things like that. DNA is a code. It's just simply a code. It's written with 4 chemical letters. Everyone's becoming familiar with those. So those chemicals begin with the letters G, T, A, and C. And there are 3 billion of these in a human genome. Okay? That's a big number. 3 billion chemicals stitched together to form a genome. And if we read that genome, we call that sequencing. Okay, so I'm going to refer to gene sequencing. It's simply a technology that allows us to read those 4 letters over and over again. Now, in preparation for this, I thought to myself, well, the human genome is getting a bit old hat now. What other genomes have recently been discovered and published? Okay, so there they are. Someone has recently published the genome sequence of the Californian 2-spot octopus. Obviously, we needed to know that, didn't we? But what's interesting about that is that octopuses have a set of genes that no other beast on the planet have, okay? And those genes are involved in camouflage. So you can imagine those genes are going to turn out to be very interesting, and wouldn't be great if we had some of those too. Okay? Especially on an event like this, I could just camouflage myself and walk out of the room. Melons, I don't actually know why someone has sequenced the genome of melons, but they have, and so there are melon geneticists who are obviously working on melons for some reason. Woolly mammoths, that's very interesting. If you sequence DNA, you can actually work out how old an organism is, and some scientists have worked out that the last woolly mammoths in northern Europe were still alive when the Great Pyramids of Egypt were being built. Isn't that interesting? It must have cost millions and millions of dollars to get that piece of fact. The North Island kiwi, its genome tells us that it has black and white vision. There's been a lot of debate about whether these little birds can see in the dark, and now we know they can. Black rice, it's boring actually, but essentially lots of different types of rice are black, and if you look at the gene mutation, the fault that makes it black, it's exactly the same in all species of black rice, and that one mutation occurred thousands of years ago in wild rice, and it was considered to be attractive. Emperors of China liked black rice, it was supposed to give them power, and so that little mutation was bred in to lots and lots of different types of rice. Okay, there we are. So that's genomes out there in the world, but there are lots of people interested in genomes, but I'm interested in the human genome. What I'm really interested in is the way in which we can use the information in your genome to dictate how we actually look after you. So there's this concept of genomic medicine. It's quite new, and it's coming at us thick and fast. So we use the information in your genome, either from stuff that you inherit from your parents, or maybe if you've got cancer actually, we can look at the changes in the genome that occurred in the tumour itself. And we can actually even look at the genome in the microorganisms that are infecting you. So one of the ways in which we'll develop very rapid microbiology diagnostics is by actually looking for the DNA of microbes in your blood, or your spit, or your wee, or something like that. Where did the word genomic come from? So we know that the word genome came from a botanist, rigorously defined word. How did the word genomic come along? It's this guy here, Thomas Roderick. He's a very famous geneticist, and one day after a meeting in Bethesda in Maryland, he and some colleagues went to a building which is now demolished actually, which is sad because this should have a blue plaque on there on the outside. This was a raw bar, okay? This is a place, very sort of Eastern American type thing. You go and eat raw fish and oysters and stuff like that. So these guys were sat around a table having a few beers after a busy meeting, and Thomas Roderick came up with the term genomics. Okay? Now if you look at what was, he was quoted later on in life, because he obviously became very famous for having invented this word. So he said, we sat around and talked. We were into our second or third picture, so that's a big glass of jug of beer, when I proposed the word genomics. I don't know exactly how I came up with the word. Okay? So there we are. That's serendipity for you. Right, slight change. First, next question. What's normal? Okay? You might think that's fairly straightforward. This is one of the most difficult questions to answer in the whole of medicine. What is normal? If you can't define what is normal, you can't then define what is abnormal. Okay? So the first question that I would like to think about is, what is a normal human genome? Now, most of you actually will remember the first Human Genome Project. It took a very long time. Okay, so it was planned in America in 1984, when it started in earnest in 1990. Okay, it took 13 years to do, and it involved 20 laboratories around the world, each undertaking a piece of sequencing to actually read a bit of the human genome, and the whole thing was stitched back together again. And between them, they managed to sequence about 90% of the whole genome. They missed out some difficult bits, which have got complicated names, but 90% of that DNA was sequenced, and that cost $3 billion. That's quite a lot of money, actually. And that was a sort of National Institutes for Health project. At the same time, a businessman set up a company a bit later on, and he started a little race, okay? And he managed to sequence his own genome. They were both published at the same time, actually, in different scientific journals. Most people in the UK will have seen these two guys. Okay, so we had President Clinton, and at the same time, Tony Blair, speaking on different sides of the Atlantic, and I think it was Tony Blair who had to get out of bed at some unreasonable hour to do this, and they really trumpeted this. So this was in the new millennium. One of the most important things in the first months of the new millennium was the publishing of the first draft of the human genome sequence. Tony Blair described this as a technological triumph. The President of America got a bit carried away, I think. Okay, he was talking about this was a very religious event for him, but nevertheless, there it was. Okay, so that was the first genome sequence. Over the years, that genome sequence has been updated, refined, purified, and made more accurate. And once something very interesting happened some years ago. Oops. James Watson, one of the original describers of the structure of DNA, had his genome sequenced. Okay? So this GRCH36 is just the sort of thing that scientists like to do. They've put a little term in there that doesn't really mean much to everybody, but that was the 36th version of the human genome sequence. Okay? And then James Watson, who's looking a little bit older on the left, and his own genome sequence done, and it was different from what we thought was normal by about 3 million of those little chemical letters. Okay? And 1.4% of his genome wasn't even present. Okay? So did that mean that he was an alien, like the Californian two-spot octopus, or did it just mean that the sequence data that we had at that point was incomplete, and that's what it was incomplete. This is quite an interesting. This is the ultimate public interaction process here. A bunch of folks decided, well, actually, what we're going to do is we're going to use more human beings and look at more genomes and try and make a more accurate genome sequence. So they took 13 anonymous individuals from a place called Buffalo in New York, and they recruited them by putting an advert in the local paper. And the advert basically says, if you want your genome sequence, come down to the town hall and we'll do that for you. Okay? So a load of folks came down and they selected 10 men, 10 women, and it looks about 80% of the sequence data came from 8 people. Okay? And one of those 8 people contributed most of the data. So by looking at all of those people, they were able to, in a way, come up with an average normal genome sequence. This is what it should look like in a normal person. And that was derived from just 20 people, actually. That was then refined in America by the 1,000 Genomes Project. So you can see now we're starting to think about bigger and bigger numbers of people. And the data from that have informed what is currently felt to be the normal human genome sequence. Now the difference here is that the folks who designed this research study tried to include as many different racial groups as possible so that we actually had an idea of what was normal across the planet and not just in Buffalo, New York. Because I would imagine, respect down to the camera here, if you're living in Buffalo, New York, I suspect that your reproductive pool is fairly limited. Okay, so the genome sequence in Buffalo, New York might be quite specific to Buffalo, New York. So you can look this up on the internet if you like. You can see how many different types of human being were included in that. But the numbers of each racial group are relatively small. So inevitably, that's not going to give us the most accurate number, the most accurate genome sequence. However, that's where we're at the moment. This project told us that in those 3 billion bases, those 3 billion chemical letters, each one of us differs from normal by 38 million letters. Okay? Each one of us differs. So if you look at the person next to you, you're about 40 million bases different to them who look fairly similar. So much of that variation is not significant. There are about a million small extra bits or deleted bits of DNA and about 14,000 large deletions. This is quite big chunks of your DNA missing compared to what we think is normal. Okay. And the awkward truth here is that every single one of us carries about 200 or slightly more rare but potentially harmful sequence changes. Okay? That's slightly awkward. So I have to share that with you today, okay? Every single person sitting in this room, walking in the street, living down south where I'm from, probably actually it's slightly different down there, every single one of us carries a bunch of these, what we would call mutations, okay? And some of those are going to predict the way in which we will live our lives in terms of our health. Some of that stuff will actually dictate what diseases we develop, what diseases we die from. Mary Berry again. Let's go back to this, okay? Now, imagine at the top there that the Mary Berry version of a Victoria sponge cake recipe is normal. That is the reference recipe, okay? So actually when you read that, you think, well, that looks pretty sensible. Actually, that's probably the recipe that I learned in home economics at school. That looks like a standard Victoria sponge cake recipe. Below that, you'll see a recipe that's been written by one of Jamie Oliver's chefs, and he's taking credit for that, and Nigella as well. Now, what you'll notice is that if you look in their recipe books, the cakes look exactly the same, okay? But look at the ingredients, they differ, okay? So Jamie Oliver actually put some rose water in his Victoria sponge cake. That's probably quite tasty. Nigella put some corn flour in, and she puts a bit of milk as well, whereas Mary Berry doesn't. These are all perfectly legitimate versions of a Victoria sponge cake recipe, but you can see that they vary, okay? And that's essentially what's happening in genes. If you think about a gene like a recipe, then many of us vary just in the same way that these sponge cake recipes vary. Occasionally though, if you look down at the bottom, you'll have a change in a gene, which is like one of these. So if you look at the bottom purple recipe, what you'll notice there is that this uses a lot more baking powder. Now I suspect this causes your cake to fizz over in the oven, okay? So that's what we call a point mutation. One single letter in that recipe has changed, and that means that it has a dramatic effect on the sponge cake. You can see on the right in blue is a deletion. Okay? That cake doesn't actually contain any eggs, so I don't quite know what that would be like, but it would be a bit dry, and it wouldn't hold its structure. So it'd be like pastry, yeah. So that's not a sponge cake, okay? So when we are looking at a genome sequence, we're looking at 20,000 recipes, we're looking at 3 billion bits of DNA, and we're trying to find the difference between the stuff on the bottom of these slides and the reference sequence. And if you're doing that 3 billion times, that's a massive technological challenge. So, two key advances are making this genomic medicine possible. And these two things are happening here in Newcastle. In fact, Newcastle has a very, very important role in the world in the development of genomic medicine. The first, at the top, is miniaturisation. And I very amusingly wrote that in very small type. And I'm not going to talk about that actually. And you may already have heard Professor Sir John Byrne talk about that type of thing. These are handheld DNA analysis devices which enable you to get a very, very rapid diagnosis by just looking at one or two of those DNA letters. The other thing that's happening here is whole genome sequencing. And that's all about the 100,000 Genomes Project. So the key players here, who've done it, okay? So sometimes when I wake up in the morning and I think to myself, why on earth are we doing this? I think of these wonderful figures in front of us. Two of our recent Prime Ministers had children with genetic diseases. That actually makes change possible when you start to see things like that happening. David Cameron wanted to prioritise the development of whole genome sequencing, the development of genomic medicine in this country, because the NHS is in a position where we are fully networked, we've had access to a population. Jeremy Hunt was basically told to go and deliver this, and his friend George Freeman, who has the longest parliamentary title I can find, he's currently called the Parliamentary Under-Secretary of State for Life Sciences. Really, really kind of trips off the tongue. These guys are responsible for coming up with the idea and starting the whole process. So the 100,000 Genomes Project is actually the Department of Health's flagship transformational project. It wants genomes, genomic medicine, to transform the way in which we deliver healthcare in this country. It's going to generate some income. So one of the ideas is that we will be the first healthcare system in the world to introduce genomic medicine and genome sequencing. And this will be the go-to place for industry that wants to come in and do research with us. And that will generate an income. And this is part of the Treasury's recovery plan, actually, from our financial crisis. NHS England has commissioned this project. It's not actually a research-funded project. The British taxpayer is paying for this project to the tune of £300 million. There's loads of big numbers here, aren't there? £300 million over 3 years. And it's the largest sequencing project in the world ever. Okay? That's cool. And we're taking part in that here. And it's not a project. I don't know why it's called a project. It's actually a service development. It's an NHS service development that's linked very closely with a research project. There's a subtle difference. I might come back to that. So the NHS England have created a company called Genomics England to run this project, and a large biotech company called Illumina have won the contract to deliver the sequencing technology. NHS England commissioned 11 genomic medicine centres to effectively deliver patient samples into the project, but those genomic medicine centres have a big role now in rolling out genomics into mainstream NHS practice. Over the next 2 years now, we're nearly at the end of the first year, 25,000 people with cancer will take part in this, and that will include a DNA sample from them, the stuff that they inherited from their parents, but also from the tumour. And what we're interested to see is how the DNA code has changed in the tumour, because some of those changes will tell us how to treat that patient better. And there'll also be about 50,000 rare disease patients. These are patients who many clinicians won't normally see routinely, but at a place like the Centre for Life, we have an expertise in people who are living with very rare diseases. And there's a focus more on diseases that are clearly affecting multiple members of the family, or if, a good example is a child who has a learning problem and who may have some kind of physical disability as well, would be interested in DNA from that child and both of their parents. And at the moment we're trying to limit the number of isolated cases coming into the project, because that, for technical reasons, that makes analysis and interpretation difficult. So by the end of 2017, in the NHS, so the NHS, you might have noticed the NHS hasn't exactly got a great deal of cash at the moment, and so its ability to do huge new enterprises like this is relatively limited. So a lot of this project is being rolled out with the sort of British spirit, if you like. And by the end of 2017, we should have delivered 100,000 genomes. These slides are just to show you the number of contracts that exist. So in typical British fashion, this project is underpinned by loads of contracts. Okay, so there are contracts around the samples that we're going to provide, the number of samples. We are intensively performance managed because the NHS loves to do that to us. There are detailed specifications about the IT systems that we need to use. There are contracts around sharing data between genomic medicine centres, and so on. I won't bore you with all of those, but I do remember going to a very, very posh legal firm in London last year to be talked through all of these. And this will have cost a very great deal of money, all of this stuff. In the middle are some unique and new structures which have got a very difficult title. They're called Genomics Clinical Interpretation Partnerships, or GCIPs. These are groups of clinicians and research scientists and folks like yourselves who have an interest in one particular disease or disease area, and who would like to have access to all of the data that we're going to generate here for research purposes. So, in other words, there's a clinical arm to this. This is about finding diagnoses for patients, but the GSIPs are there to help coordinate research in those patients who don't get an answer. We also have a Genomics Education program within this country. So Health Education England, an NHS structure, has a Genomics Education theme, and that links in with what we're doing as well. Today is part of that. It links with both PPI activities like this, but it also links with a new master's program that Newcastle University has developed around genomic medicine. Not everyone can take part in this. This is specifically limited to a certain number of rare diseases and cancers. Lots of different rare diseases, but nevertheless it's quite limited, because effectively what we're doing is trialing this to see whether it's effective. There are very complex data models around these, so as well as giving data, it's giving blood samples, patients also are going to be giving us loads of information about themselves, their healthcare problems and so on. It all combines down into a rich but very, very comprehensive data set. Someone taking part in this will give consent. They have to agree to this. And they will consent to a variety of different things. Firstly, to having their DNA sequenced. Secondly, to the storage of data about them. And thirdly, to potential future research. And they're specifically consenting to these types of things. Testing to try and find the cause for the disease in question. So that makes perfect sense, I guess. If I've got an inherited muscle problem in my family, I want to know the genetic cause of that inherited muscle problem. But you can also know an awful lot of other stuff. You can imagine that if you want to answer that question, you would look at a set of like maybe 1,000 genes, but there are 19 other 1,000 genes as well that contain lots of information about you. So you can also then elect to be told whether firstly you have a genetic change that may have reproductive implications, so it may have an implication for your future children. And you can also elect to be told whether you have any incidental serious findings. So as well as being told about my muscle disease, I can ask if I will be told whether I have a mutation in a breast cancer gene or a cholesterol gene, for example. Okay? So some folks feel that maybe we're opening a can of worms here, because this isn't just a simple diagnostic test. This actually contains an enormous amount of information that is of potential health importance. Every single person so far who's taken part in the study in this region has said yes to that particular thing. Okay? Because there is a sort of anxiety, I think, about genes generally, and everyone's becoming more familiar with the concept of cancer genes, for example. You have Angelina Jolie's life to look at in the newspapers every day, and we understand the impact of all of these other genetic changes. There is part of the project which is currently closed. You cannot say yes to this at the moment, and this is expressed in this term, and any other unexpected findings. Okay? So buried deeply within those 3 billion letters, there will be very rare changes in genes that we don't fully understand yet. Okay? And in the future, it is believed that we will be able to ask people whether they would like to know if they've got one of those. Okay? Now that I would propose today, we can debate this, I would propose that that really is opening a can of worms, and that we aren't quite ready for that. But for that reason, you can't ask for that at the moment. Folks will give a blood sample. Actually, they'll give us quite a big blood sample because we want some DNA from them, and then we want some other stuff as well. And most of that stuff is to store for future research. All of that is processed here in the Northeast, and the NHS Blood and Transplant Service are used to transport all of those samples down to a biobank facility. And the DNA samples are sent on to one facility in Cambridge, which is a bit like a factory containing things like this. So this these look like something from Currys or PC World, okay? But you can't buy one of these in Currys or PC World. This is the Illumina High-Seq 10 system, okay? So down in Hingston, there is a big factory that consists of loads of these things, and these are DNA sequencing machines. And they're quite groovy if you like stuff, if you're a bit nerdy like me. These are fantastic. So this set of 10 here can deliver 18,000 whole genomes per year. That's 50 genomes a day. So take your mind back. The first genome took 13 years. Okay, cost $3 billion. These machines can do 50 a day, and actually that array of machines there costs £1 million. So it's about £1,000 a genome, and actually each genome is read at least 30 times, okay? And that gives us really accurate data. So that's pretty cool. How's it done? Basically you break up the DNA into lots of different fragments. All of those fragments are sequenced separately, and then they're all lined up back again with each other, okay? And so you can see, so if you look at it on the top, it's an unpronounceable word, isn't it? I always thought it'd be really good if I could learn the human genome sequence and speak it. Okay, so I could say to my sister, but it doesn't work like that. Look, if you read along the top and then look at all these things, can you see that there's a red C here, okay, which used to be a T? That's what we're looking for. There's a letter change. So, okay, so this is a what do we do? We put some baking powder in. We put a little bit of baking powder, but the C means put a whole bucket full of baking powder in. Okay, so that's what we're looking for. And imagine 3 billion of those letters. You need some pretty fancy computers to tie all this together and to look for those changes. But those computers exist. And effectively, what we get back is an extract of all of that sequence. We don't get the whole sequence data back. That would be really difficult for us to use in NHS practice. But we get a filtered view of that sequence. Okay? A computer basically tells us what has changed, and of those changes, which ones are significant. And we'll verify those in our laboratory just down the road, and we'll actually issue a report back to the clinician who's looking after that patient. And what we're hoping, what we anticipate, is that many people who don't currently understand why they've got a disease will actually finally have an answer. How are we doing? So imagine you've got colon cancer, and imagine that you consent to genome sequencing. Okay? This will become routine at some point. I don't think it'll become routine just yet. You can have it done as part of this project, but maybe in 5 years' time, this will be a routine thing. So this is just made up completely, but this might reveal that you've got 3 acquired changes in the DNA in your tumour, and those are going to guide your therapy and increase your survival chance by 40%. Some of this stuff already happens. The results may also show that you've inherited a change in a cholesterol gene, which causes high cholesterol levels, but it's very easily treatable. You may have also inherited 2 DNA mutations that increased your risk of dementia by 20%. And I ask you, would you want to know that? The information is potentially there. Do you want to know that? Some of you are saying, yeah, actually, yeah. I'd like to know that because maybe I'd like to go on that trip to Australia before my dementia starts. And interestingly, there may be a mutation there that will help decide your dose of warfarin if you ever need it. Okay? So there's information there which will dictate your response to treatment with drugs that we use every single day, but perhaps in an ill-focused way, and some of that genomic information will tell us exactly how to tailor your dose and your therapy. Okay? And we're not far from here. I used to stand up so I've been a consultant now for nearly 15 years. When I first started, I would say things like, yes, it's around the corner, or within 10 years, and that was all a bit vague and finger in the wind. And in fact, genome sequencing wasn't even really an option, or even a possibility when I started as a consultant. But now it is, and within 5 years now, I feel confident that within 5 years, a lot of this information can be extracted from you. And I ask one challenging question, and that is, are we as a nation, are you and your friends, families, colleagues ready for this? Because this is big stuff, actually. The goal is that by the end of 2017, it's a big goal, this, and it's a bit hopeful, that we'll have routine diagnostic genome analysis in 3 weeks. Okay? Not 13 years at $3 billion, but 3 weeks for less than £1,000. If you think that's crackers, I read a paper that was published a couple of months ago where an emergency genome had been requested in America, and it was done within 36 hours. So this was a child who crashed into the emergency room, obviously had some kind of genetic problem, also had a blood cancer, and the genome gave the answer within 36 hours. That's quite interesting. It's very American, isn't it? So genome sequencing is here. It's not just here generally in a vague way. It's actually here, and it's happening here in this city and in this region. We're really honoured to be part of this. This is very exciting stuff. It's deeply challenging, but it's being used for us NHS patients here in the north. And here's my question, are you ready for it? I would place genome sequencing up there with putting a man on the moon, inventing insulin. Einstein maybe, he's pretty clever. I think genome sequencing is more clever than Einstein. Anesthetics, organ transplantation, microbiology, music, computer science. It's up there with all of these things. And we've all got the chance here to take part in that.
Thank you so much. I mean, that was just absolutely fascinating. I do think it's one of the most exciting areas, period, and to have it here in the region is just fantastic. So, an opportunity for some questions now, before lunch. No, that's fine.
Robert
Yeah. Just speak up, Robert.
Well, I'll speak up. I've got quite a loud voice. Well, someone comes in where we've already got through diagnosis procedures, like for instance here in the northeast we've got the colon cancer check. When that person comes in the future, will you have to sequence the whole DNA, or will you be able to pick out the pieces which are colon cancer related and do them even faster?
Could I just check everyone heard the question? Could you mind repeating the question, Paul?
Yes, I think the idea is if you come in through screening, so we have a bowel cancer screening program. If you are picked up to have bowel cancer, can we just have some focused genomic testing rather than read your whole sequence? And the answer is, yeah, we're doing that already. Okay. But we're looking at specific bits of your genome. Okay. And I think the strategic direction is that genome sequencing will become so rapid and cheap that you will just do that. Now this is going to be a piece of work for our DEC to get their hands on. DEC have recently been involved in a very interesting project to look at a way of diagnosing an inherited cholesterol problem. And what they've done is helped us to look at the difference between two different approaches, two different technologies. And those technologies are, do you actually just sequence that gene and a couple of other genes, or do you look at focused areas within those genes? And there's a massive difference between what is possible and what actually is clinically useful. So people like the DEC are going to help us sort those out. As you can imagine, 20,000 genes, that's going to keep them busy actually for many, many years. But I think actually in clinical practice, the very rapid turnaround test is going to be the targeted one that looks at specific things.
Thank you. Are there any other questions? Question over here. Is the microphone working, Alex? Is that? You might want to get ready to cover your ears again.
participated in the 100,000 Genome Project. My background's computer things and systems analysis. Now, the GSIPs, term I've never heard before, I do know that for my particular condition there is a GSIP in existence. I've questioned the people who are basically running that project. They did say, yes, sharing data is a major factor in this, but because I'm like that, they were very, very, very woolly about protocols on data. I don't mean security things, I don't mean ethical things. They just seemed to me, it came across to me that this GSIP, there were already genes identified for my particular condition, but they seemed very, very focused on that subset of genes. And from what the impression I've got here with the decks, etc., it's a much wider, you're looking at things in a much wider way. And my worry is that, yeah, my blood samples results sequence, when it's passed on, I've just got this nagging doubt in the back of my mind that there will be information in that sequence which will not be looked at by a GSIP because they are focusing on what they already know. I think that from what I picked up, I think it's the deck that is missing from that equation.
Thank you for your question.
That's an interesting question. How do I answer that? NHS England launched off a whole series of initiatives without, understandably actually, without having been able to define exactly what they should look like. Yeah. So the GSIPs, for example, was a concept, and NHS England said, right, there's the concept. Can you all go away and have a think about this and tell us how it works? And the different GSIPs for different disorders have evolved in slightly different ways. Their role is to define exactly which genes we look at first of all. Okay, so in truth, your genome sequence that will be analysed by extracting out of that 30 billion bits of information a set of information. Okay, and those will be genes that we already know about. So that's kind of what you're saying. Obviously, that set is going to increase over time because new discoveries will be made, and so we would want to look at more and more genes. And there is currently a proposal that every single year your genome sequence will be recurated. In other words, that those extra new genes will be reanalyzed. That exists in digital form. It'll be on a computer somewhere, and you can just pull out the new data. The GSIPs themselves will contribute to that because they will use your data to try and find those new genes. But they're very much disease focused. I don't know if that really answers your question, but I think that what we've been asked to do here is collectively jump off a cliff together, and we're learning to fly. I can't answer your question because the answer doesn't exist yet.
I think that's what is exciting about involving patients and public in that debate at an early stage as well. I'm conscious of lunch, but I think this is a great opportunity to talk to Paul. So just one more question.
Could I just make a comment on that?
Sorry, Phil, could you use the mic just because there's people coming?
I'd like to comment on that because you have actually mentioned it, that part of the initiative of Genomics England and what we're actually doing in Newcastle is we need to train the healthcare professionals as well. And we have a master's course that has started this year as well. But just what you've actually asked, part of this is they've already, across the NHS, they've identified a cohort of healthcare professionals that need to begin that process of training how to assimilate the data that this is going to be used. And I think that's another way of actually looking at an answer to that question, isn't it?
Yes, we need actually within the NHS a new group of professionals called bioinformaticians. So these are people who can actually look at genome data and understand what it means. And the doctors who are using these tests also need to become genome savvy as well, because otherwise they're not going to know what these results mean when they land on the table. It might seem trite, but I can remember as a junior doctor MRI being introduced, so magnetic resonance imaging, and there was a very uphill learning curve for MRI. So when you got a result from an MRI test, you needed to be trained how to analyse and to understand those results and to use those in clinical practice. So that was a very powerful new technique that could look at a human body in immense fine detail. And it started to reveal all sorts of things that we'd never really seen before. Now we understand that they're just normal variations, but then we didn't. And we're on a similar learning curve actually with genomic medicine, and we've got the same challenges. We need to bring everybody up to speed. But there's also a social component to this as well. I think that as a society, we've got to start getting a bit more comfortable about genetic information. There have been some concerns expressed about, okay, who's got access to my genome data? Okay. You can actually tell where someone's from their genome data. Okay. You can look for population-specific changes in your DNA. So you can find all sorts of stuff about people, and we've got to get all this right. Okay.
Thank you. And I think, today is really the start of our contribution through DEC, through partnerships with the AHSN, the BRC, the BRU that John mentioned, to really sort of have that and widen that public dialogue out. We're going to hear from Andy Hall and colleagues later about some of the important work that they're doing and the work around ethics and consent, etc., which is so fundamental. So if you have questions, again, when you reflect, send them in. As we always say, we collate them, we'll pass them on to Paul, to the people, and we'll look at setting up future events which might be a little bit more focused, perhaps on ethics, perhaps on responding to some of the questions that came through, because that partnership is really ongoing, and it's just such an incredibly, it feels like you're at a frontier, doesn't it? I mean, it's just fantastic. So massive thanks, Paul, for that fantastic presentation, and really, really appreciate it.